Thank you, Yves, for introducing. As you may guess, this is actually a rather technical topic, but what I will try to do is to give a metaphoric presentation of this topic instead of talking too much in technical details. So we've already heard that the inheritable genetic information of any organism is encoded in the DNA, which is distributed on a number of chromosomes. And the information in the DNA can be described as a series of letters, much like the text of a book. The information in that DNA is a large package of instructions to assemble and manage the living organism in which it is stored. So let us take this metaphor a step further. In nature, the information in the DNA of two individuals are never identical, as we also heard earlier this morning. So there are numerous small differences across populations, and new differences develop, for example, as a result of environmental exposure, what we often call spontaneous mutations. Such mutations may be small, like single character substitutions, or they may be larger, for example, insertions, deletions, rearrangements such as duplications or inversions. Genetic modification is a tool, or more correctly, a set of tools that are used to change the genetic instructions in an organism. In the GMOs hitherto put on the market, this has mainly been done by inserting new extra information. However, with new technologies, it is also possible to delete information or to specifically change single characters. This is important because these new techniques introduce new technical challenges to GMO detection. So gene modification technology itself is gradually turning into a text editing rather than an engineering technology. Now, let us go back to the metaphor, the text of a book, and let us take a well-known example, William Shakespeare's Hamlet. This is a play and therefore holds instructions for the scenes, the entry and departure of characters, and the specific dialogue. Evidently, some of the text motifs appear several times, while others appear only a single time. Furthermore, there are fewer combinations of characters and words that do appear in this text than are absent. Examples given here. A genetic modification is comparable to a desired change to the original text, such as insertion of a new sentence to the dialogue. Always look on the bright side of life from Monty Python's Life of Brian would perhaps appear misplaced. But what if the sentence was deliberately placed in your copy of Hamlet because the publisher felt that the play needed a touch of ridicule? Now, returning to the detection issue. How do we detect changes in the genetic information? In the course of the last 2 decades, the prevailing analytical tool has been the polymerase chain reaction, which has also been mentioned earlier, and I do not intend to explain it to you. However, PCR is essentially a tool that can detect very specifically the adjacent presence of 2 specific words in the text in a specific orientation. So if you know what you're looking for, perhaps such as always look for the on the bright side of life, you can use always and bright in that particular orientation, and you will detect the insertion. Unfortunately, the text we study is not the equivalent of a single copy of the book. A food or feed sample typically contains material from hundreds or thousands of copies of the individual organisms, such as single plants of the same species, of which some can be GM and some can be non-GM. In our metaphor, this corresponds to hundreds or thousands of copies of Hamlet coming from a large but unknown number of publishers, and each edition is likely to be slightly different at the detailed level despite the majority of similarities. And some of those minor differences have the potential to affect the detectability of targeted text elements. Now, I have to talk a few words about numbers. The text of one page typically corresponds to approximately 4,000 characters. In comparison, the full set of chromosomes in a plant cell corresponds to between 600 million and 200 billion characters. A typical GMO test contains DNA from approximately 50,000 cells, which is the equivalent of between 30 trillion and 10 quadrillion characters. And yet, the typical target of a GMO test, this is a sequence of approximately 100 characters, is detectable in as few as 5 copies and can be quantified at concentrations lower than 100 copies. So we must conclude that PCR is extremely sensitive. Food and feed samples are never really pure. There is always some degree of botanical impurity in plant-derived products. In a bag of wheat flour, it is likely that an analyst will be able to trace DNA from other cereals crops and a considerable number of weeds too. If you look at an agricultural field, you will see that weeds as well as food crop plants of other species commonly appear in between the plants seeded that season. Botanical impurities can also be introduced during harvest, transport, storing and processing. In our metaphor, the botanical impurities correspond to unintended presence of the text of other books in the sample that we analyze. Chance is that if we detect a particular text element suspected to be the genetic modification, it could be present in a text other than Hamlet. So perhaps what we detect is not a genetic modification, or it is a different genetic modification than we are looking for. Thus, we have to discriminate between putative insertions and true insertions. This is simple if the text inserted is a construction that has never before been introduced, and we see a fusion of text from two unrelated texts. That is really a tag for unique identification. Unfortunately, I have to blur the image further. When the food or feed product is processed and composed of several ingredients, two important things happen. First, the processing leads to fragmentation of the text. In our metaphor, it corresponds to cutting the contiguous text of the book into shorter blocks that behave independently in the space of text elements. Secondly, the specific plant species in consideration is less dominant, and the complexity of information in the sample considered is much larger. In our metaphor, this corresponds to searching for the specific text element of the genetic modification in a collection of very divergent texts. Perhaps the collection even includes the original source of the genetically modified text, or perhaps the construction we believe is specific to our genetic modification has been inserted in more than one book. Thus, we need to find a way to discriminate between copies of the target text in Hamlet and other texts. So I would like you to take note of the differences between the motifs highlighted in yellow and green respectively here. Finally, we need to know if a particular species is present. In our metaphor, that means whether Hamlet is present in our sample of text and eventually how many copies of Hamlet the sample contains. This number forms the reference for determination of the quantity of genetically modified Hamlets in the sample. The labeling regulation for GMOs in the EU requires that this is expressed in %. So, we choose as our target for detection identification of Hamlet a text unique to Hamlet and present only once in each copy of the book. To be or not to be, that is the question. The estimated quantity of our genetic modification then becomes the number of times the genetic modification, to always look on the bright side of life, is detected, divided by the times the reference to be or not to be, that is the question, is detected, and we need, of course, to multiply by 100 to obtain a %. At this point, I need to be slightly technical and visualize the insertion of a genetic construct into a GMO. The resulting DNA string contains a series of alternative targets for the PCR detection methods. Each of these has advantages and drawbacks for GMO detection, identification, and quantification. Level 0 are the species-specific targets, to be or not to be. Suitable for screening to detect and identify species. Basically, we ask the question, do we know of any GMO belonging to this species? If the answer is no, then we do not need to continue. If the answer is yes, then we have to continue our search.